I'm Rebecca Cook. I'm an associate professor of cell and developmental biology in the School of Medicine at Vanderbilt. I have an associate's appointment in the Department of Biomedical Engineering at the School of Engineering. I do breast cancer research, and that can be divided into three main pillars. Part of my research is understanding how the breast normally develops and how protein signaling pathways that are involved in breast development get commandeered by cancers, and how we can use that information to treat breast cancers. We're also looking at the tumor microenvironment of breast cancers and understanding how the immune system plays a role in the development of cancers and how we can target the immune system and retrain the immune system to attack cancer cells. We use a variety of tools. We've really adopted a multidisciplinary approach. One of the main tools that we use in our lab is cell culture, which allows us to culture breast cancer cells and manipulate them to express specific genes or proteins, or to lack specific genes or proteins. That allows us then to understand and interrogate the processes that they are involved in. Another method that we use is we use mouse models of cancer, both genetically engineered mouse models of cancer, to really understand how tumors develop within their native context of the tissue architecture. And we use transplantable tumor models that allow us to really examine differences in human tumors and versus mouse models of cancer, for example, and how each might respond to different experimental therapeutics. So I was originally interested in developmental biology. That's what my original training was in. And that's the study of how embryos develop from a single cell into this fabulous multicellular, multifunctional, multi-organ human. And from there, it became increasingly apparent that cancers often use the same genetic and protein signaling pathways that developmentally regulated processes use, only with different outcomes. And so there's this interesting interface between normal physiological development and cancer progression that I wanted to study. And using that platform, I had a unique perspective on how we can treat cancers specifically. I came into breast cancer research when I came to Vanderbilt and started my training with Carlos Ardiaga, who's a world-renowned breast cancer clinician scientist. And under his training, I was really able to expand the idea that these signaling processes that control development in the breast are used by cancers, and we can target those specifically to treat the breast cancers with really great specificity and with very little collateral damage to the rest of the body. And so that became my area of expertise. That journey became much more personal when my husband's mother, who I had really just adopted as my own mother, she was battling breast cancer for nearly almost 23 years before she lost that battle. And watching her in the final years of that battle, the grace that she conducted herself with, and the strength that she had, and the support that she gave me for what I was doing really inspired me to carry on that research after she passed. Basic science is critical for what I do. These are asking and answering the questions that lie at the core of improving outcomes for patients with breast cancer. These are going to help us understand why breast cancers occur. And if we can figure that out, then maybe one day we can prevent breast cancers. These basic science questions are going to be able to help us detect breast cancers much earlier, which is also going to save lives. And these basic science questions, and their answers are going to find cures that right now don't exist. Scientifically, we've made a lot of discoveries that we've really fought hard for. And these include understanding why women diagnosed with postpartum breast cancer, why these women are so vulnerable to this lethal disease. So we studied that and found that the natural processes that occur in the breast during postpartum involution, when all of the milk-producing epithelial cells of the breast are removed, that produces a wound healing response in the breast, which suppresses the immune system to decrease inflammation, but this allows tumors to escape immune surveillance. And so our discoveries demonstrated the mechanism by which that occurs, and we've proposed an experimental therapeutic regimen to address that. That's been a really rewarding process for us. I mean, think about the health cost, the emotional burden, the societal costs of a young woman who just had her family and she's diagnosed with breast cancer, and knowing that that disease is quite lethal. So it's been a really rewarding process to discover that at its outset and see it through to a potential therapeutic. I am strictly basic science, but I have a very translational angle on it. My science, my research has been moved into clinical trials, but at that point I generally become hands-off with it and wait to, and eagerly anticipate the results of the clinical trial. So those trials are currently ongoing. So for example, we have a body of research that studies the role of a receptor tyrosine kinase called ErbB3 in estrogen receptor positive breast cancers. And our research suggested that combined targeting of ErbB3 with an endocrine therapy would result in superior outcomes compared to targeting with either agent alone. And those studies are now in clinical trial based in large part on the work that we did here in this lab. So I'm thrilled to see that that's in clinical trial and anxiously awaiting the results of that trial, but it will be several years before that comes to fruition. We have ideas and we have passion for these ideas, and we want to explore these ideas, but without the funding, these ideas will never be supported, never explored. So we rely on funding. We gain funding through different sources. Some of it is taxpayer-based funding. So, for example, grants that we apply for, and if they're awarded through the National Institutes of Health, for example, those will help pay for what we do. Or we could apply to funding from private institutions, for example, the Susan G. Komen Foundation. And the foundations are dedicated to supporting our research. Without funding, we wouldn't be able to do this research. Really, the key cost that funding supports are the people that work in this lab. These are people that are driven to find answers, to find cures, and their salary is supported by these grant dollars. Otherwise, without these grant dollars, we wouldn't have money to pay our people, and we wouldn't have people to do our research. And so we rely on funding to keep the machine moving forward. And so I've had the opportunity to train over 20 undergraduate students. I've trained 4 graduate students, and I've trained 4 postdoctoral fellows. And each of these individuals have risen to and exceeded their expectations. They've each achieved great successes, publications, discoveries, awards. They've been honored by being invited to give presentations of their research across the country and even all over the world. which makes me proud as a professor, as their mentor, guiding them through this process. I would say follow your passion, because that passion will drive you on days when science isn't going your way. That passion will drive you forward. Another piece of advice is never forget why you're doing this and who you're serving. A lot of us, including myself, get into biomedical science because we want to make a difference. We want to help people with our skills, with our love for science. And there are days when it becomes really easy to separate yourself from that original motivation. But when you look around and you see the people that you are helping, the patients, the clinicians, the people in your lab who you're mentoring, the people who teach you things, you are providing a gift, a service to the people around you, and that can be a really motivating factor. And the last piece of advice I have is to never stop trying. Never stop trying. If you can't find the answer to an experiment, to a question, one way, try another way. Keep trying and never give up. Attracted me to Vanderbilt as an undergraduate was the high-profile allure of a well-established, well-known university that maintained a small community atmosphere. When I got here, I was really impressed by the quality of the education that I received, which I honestly didn't fully appreciate until I left Vanderbilt and was able to compare my college education with the education that I was seeing around me. But Vanderbilt was so dedicated to the students when I was an undergraduate, and I can see that that's still the trend 25 years later. I went to University of Cincinnati for my graduate work and came back to Vanderbilt to do my postdoctoral training. I trained with Carlos Arteaga. I was thrilled with Vanderbilt because of the close interaction between the university and the medical center, which allowed us to do basic science research in a very translationally patient-oriented way. That was very attractive to me. We had state-of-the-art facilities. We had good administrative support all the way from the chancellor down to the people who helped us submit our grants. The framework and the architecture were there to support the success of each individual investigator, which was really attractive. I left Vanderbilt for family reasons to go to University of North Carolina while my husband finished his residency. And when he finished, he said, "You can go anywhere you want. I'll practice medicine wherever you want. Where do you want to go?" And I said, "I want to go back to Vanderbilt." And so we've made that happen, and that was 11 years ago. I've been here since, and I know it was the right choice. The collaborative environment at Vanderbilt is unparalleled, and you'll hear other investigators at Vanderbilt say the same thing. It is easy to collaborate with other individuals at Vanderbilt. It is open, it is supported, and it's easy to be successful at Vanderbilt. We work really hard, and our work is rewarded. So that's a really good feeling when your efforts can be rewarded.